Now that you've made a transcript, you have to figure out what part of that transcript you actually want to transcribe to make a protein. The way DNA is set up, it's not ordered at all. You could have a gene for your eye color next to the gene to make a muscle protein. How does all of that disjointed material become one very clear transcript for one very clear protein? Alternative splicing! Imagine you had to write a paper with a 500-word limit and you're at 900 words. You have to cut down 400 of those words to be able to make it into your limit to submit your paper. The same thing is happening with alternative splicing. You're not going to be cutting the same 400 words every single time, but you're always going to end up with some type of coherent paper that you can turn in. The genetic material that gets removed will stay inside of the nucleus as something known as an intron in the nucleus. The things that exit the nucleus are the exons, and that is the actual material that is translated into making a protein. How does this translation occur? I'm glad you asked. Once the full exon has been made, similarly to how DNA gets a tag of the TTA-GGG added to the end by telomerase to prevent degradation during replication, there's going to be a cap that's put on the 5' side of an RNA transcript, as well as a poly A tail or a whole bunch of adenine nucleotides added to the 3' of an mRNA transcript to prevent degradation. Now that there is a cap and a poly A tail on the mRNA transcript, it is considered mature and it is able to leave the nucleus to go find a ribosome. What's a ribosome? The place where proteins are made. The way a ribosome is set up, there's 3 different sites inside of it, the molecule that comes and adds the amino acids, to be located. The first site in the ribosome where a tRNA enters is called the A site, or the anchor site. When it's starting to anchor into the ribosome, it enters through the A site. The structure of a tRNA is a combination of RNA and a protein because that RNA is going to have to bind to the mRNA transcript. However, it's not as long as an mRNA. It's only 3 nucleotides long. The 3 nucleotides that's on a tRNA is known as an anticodon, and it will bind complementary to the codon that is on an mRNA transcript. The first codon that has to be read is AUG. However, the tRNA molecule is not going to have AUG. It's going to have UAC bound to it as the anticodon. Will be attached to a methionine amino acid. So the first amino acid that you will see on any peptide chain is a methionine that comes from an AUG codon on an mRNA transcript. Once that tRNA comes in and anchors, it's going to move into the P site, P for peptide bond. So the P site is where that amino acid is now being able to be bound to all of the previous amino acids. The tRNA molecule doesn't need to be there anymore and it can exit. So after that peptide bond is formed, the tRNA molecule, which has lost its amino acid and is now called uncharged, enters the E site of the ribosome, where it exits the ribosome to go find another amino acid to restart the process. The A site of a ribosome is for the anchoring, where it begins. The P site is where the amino acid binds via peptide bonds to the other peptide chain. The E site is where an uncharged tRNA molecule exits the ribosome to go find another amino acid. Because this transcript comes from DNA, mutations in this transcript can lead to 4 different types of protein mutations. 1, it stops early and you have a truncated or shortened protein. 2, you're just replacing one amino acid with another amino acid. 3, you remove one nucleotide and completely shift all of the codons that are being read thereafter. Or 4, nothing. It's a mutation, but that mutation still gave you the same amino acid, so you don't see anything different in the protein function. When a mutation leads to a truncated protein, it's called a nonsense mutation. When a mutation leads to switching out one amino acid for another one, but the rest of the protein stays the same, it is called a missense mutation. When all of the codons have been shifted or the reading frame for the codons has shifted, this is called a frame shift mutation. And when the mutation leads to the same amino acid being made, which means the same protein is going to function, it's called silent. How could you have a silent mutation occur in the first place?
Wobble, baby, wobble, baby, wobble, baby, wobble, yeah. Wobble, baby, wobble, baby, wobble, baby, wobble, yeah. Wobble, baby, wobble, baby, wobble.
For the uncultured, that's the wobble dance. And wobble theory is how multiple codons can code for the same amino acid.